Welcome. We completed already about the pre-fertilization events and also fertilization events in the flowering plants. Now we have come to the last stage, that is the post-fertilization events in flowering plants. So now in this video I'm going to begin with the post-fertilization events in the flowering plants, that is angiosperms, and we shall discuss up to the development of embryo in the dicotyledonous plants. Right, so let's begin this. Now let's see about the post fertilization events. Okay, so in this I'm going to discuss about the structures and also the events. Right, so we studied already the definition of what is actually the post fertilization. Right, so whatever that events after the formation of zygote in a sexually reproducing organisms, okay, so are all called post fertilization events. Right, so whatever that events that we are going to study after the formation of zygote, right, they all will come under this post fertilization events. Now let's see what are those post fertilization events in case of flowering plants. Right, the post fertilization events in flowering plants, so those are nothing but angiospermic plants in angiosperm. So let's see the post fertilization events. What will be those post fertilization events? So this one, it in fact it includes the endosperm development. So there is a endosperm development, and then we will also see the embryo development. Of course this embryo will be developed from the zygote. So the endosperm development, okay, embryo development, and then the maturation of ovule or ovules into seed. So after the fertilization, what happens is the ovule will develop into the seed, right? And then we will also see that the maturation of ovary into fruit. Okay, so the maturation of ovary into fruit. So these are all the post fertilization events that will be occurring in case of flowering plants, that is angiospermic plants. Right, so what are all those? The first one is endosperm development, then embryo development, then maturation of ovule into the seed, and ovary into the fruit. So in our previous videos also we have seen that the ovule is going to develop into the seed after the process of fertilization, okay, and the ovary will become the fruit, and whatever that zygote is going to develop into embryo. So that let me mention again here. So here that primary endosperm cell, that what I have told in the last video, that primary endosperm cell is going to develop into endosperm. Right, so that's what we have seen here. The next one, embryo development, where the zygote is going to develop into embryo. Right, and here the ovule, which I mentioned already, that is going to develop into seed, and then ovary will become fruit. So these are all the post fertilization events that we will be studying in flowering plants, that is angiospermic plants. Right, okay. So now let's see these things little detail. So right in the last video I told you that, so this is the embryo sac, of course, after the fertilization. So now I'm calling it PEC, the primary endosperm cell, and this is the nucleus of that PEC. So this stands for the primary endosperm cell. Okay, and whereas this one it is called PEN. So this one stands for primary endosperm nucleus, stands for primary endosperm nucleus, of course. So these are actually triploid in condition, right? And this is the micropylor angle. Okay, and opposite we know this is the chalazal end, and we know that the zygote is formed, okay, on the micropylor end. So this is the zygote, right? So now in majority of angiosperms, what happens is the endosperm will form first, okay, and then later the zygote starts dividing to form the what I call embryo. So of course some amount of endosperm is formed, the zygote is not going to divide. So in other words, I say that endosperm formation takes place before embryo development. So here in this case the zygote will remain inactive for a certain period of time, okay, until amount of right, so certain amount of endosperm is formed, right? So why is this condition right arises? Because this will ensure that zygote will get assured nutrition, okay, then which can help the zygote to divide and result in the formation of embryo. So this zygote is going to divide by mitosis and results in the formation of embryo. I hope we know zygote is diploid, so hence the embryo formed will be also a diploid. Okay, so as I told that the endosperm formation, okay, will occur before the embryo development, or before the zygote starts dividing and resulting in the formation of embryo. Certain amount of endosperm should form, right? So this will help in, so this condition helps in assured nutrition to the developing embryo, right? So helps in assured nutrition to the developing embryo. So that what we call it, okay. So that is the, in fact, the reason why the endosperm formation takes place before the embryo development, right? So now let's look at this endosperm development first, then later we shall see how the zygote is going to develop into embryo. Okay, so now the primary endosperm nucleus, right? This primary endosperm nucleus is going to divide, okay, and results in the formation of many nuclei. So it results in the formation of many nuclei. So in this case, all these divisions are called free nuclear divisions. I hope you still remember what is the meaning of free nuclear divisions. Means those divisions which will be not followed by cytokinesis immediately, right? So this is what I say, the free nuclear divisions. So the primary endosperm, okay, nucleus divides, okay, of course by mitosis, and results in the formation of many nuclei. So the pen divides by mitosis and results in the formation of okay, many nuclei. So that results in many nuclei are formed. We know this primary endosperm nucleus is a triploid. So hence whatever that okay nucleus formed will be also triploid. So all these nuclei which formed are triploid in condition. Right. So this is what I say, the free nuclear endosperm. It's called the free nuclear endosperm. So the free nuclear divisions are immediately not followed by the cell wall formation. So hence there will be no cellularization taking place immediately. So what you see is a lot of nuclei formed. Okay, so such type of free nuclear endosperm you can see in case of coconut. Okay, so you will see this free nuclear endosperm. So that is what I call the coconut water, what we drink. The okay, the coconut water what we drink is actually the free nuclear endosperm. Right, of course after some time what happens is after this free nuclear endosperms, right, I mean the free nuclear divisions, it results in the formation of a cell wall. So you can see here there is a cell wall formation, right, and as a result the free nuclear endosperm, right, slowly changes to, right, the cellular endosperm. Right, so you can see here this is, right, the cellular endosperm slowly results in the formation of the cellular endosperm. Right, so this is the cellular endosperm. So this also you will see in case of coconut. Okay, that white kernel that surrounds the, okay, the free nuclear endosperm is nothing but the cellular endosperm. So this is the white kernel. So this is the white kernel that is present, okay, around the free nuclear endosperm. So that's what I say, the cellular endosperm. So here the point is that after the nuclei divide, so I told that those divisions are free nuclear divisions, right? And so this mitosis in fact is a free nuclear divisions, which is not followed by the formation of cell walls immediately. Of course it will take place little later, and you can see the cell wall formation starts from outside and slowly moves towards the inside. So that's why you will see, the white kernel all around the free nuclear endosperm in case of, okay, coconut. So this cellular endosperm that you will also see in case of coconut. So this also you will see in case of coconut. Okay, uh, now so of course this later after the formation of cell wall, the formation of cell wall, it results in the formation of a triploid endosperm. So that results in the formation of endosperm. So of course that is triploid. Now the cells of this endosperm will be having a lot of reserve food materials, or they are filled with reserve food materials. Okay, so I say the cells of endosperm are filled with reserve food materials. So that's the okay, point here which will act as the nutrition, or I say the food to the developing embryo. So now once a certain amount of right endosperm is formed, then the zygote starts, okay, dividing, and this will use this endosperm, okay, because this is the nutrition, okay, to this zygote developing into embryo. So it will use this endosperm, and it starts dividing and slowly develops into embryo. Right. Now, by the time this zygote divides and results in the formation of embryo, the zygote, okay, or I say the embryo may completely use that endosperm. So hence, okay, by the time uh, the ovule becomes the seed, there will be no endosperm present in the, okay, the seed. So depending on this, right, we shall see two conditions, right? So let's see endosperm. So this endosperm, right, is completely consumed by the developing embryo. So it may be completely consumed by the developing embryo, right? So if it is completely consumed by the developing embryo, right, so in this case you see that there will be, okay, so there is no endosperm, so there is no endosperm in the mature seed. So you don't find the endosperm in case of the mature seed. Okay, so that's what we see if that what I call the endosperm is completely consumed by the developing embryo. Okay, so as the seed is not having any endosperm, I can call it non-albuminous or ex-albuminous, which I'm going to discuss this under the seed. So in the next video I will discuss about that. Okay, now in another condition, what happens is the endosperm is not completely used by the developing embryo. So I can say incompletely consumed by the developing embryo. So the developing embryo did not use the endosperm all. Okay, so hence in the mature seed we will be seeing, okay, some amount of endosperm still persists. Okay, so there is some amount of endosperm, okay, still present in the mature seeds, right? So that's what the endosperm, the fate of the endosperm here is that it may be completely used by the developing embryo, or it may not be completely used. Okay, so hence in this case when it is not completely used, we will find some amount of endosperm which will be still present in the mature seeds. Of course, okay, this endosperm, okay, which is leftover endosperm, that can be used up during the seed germination. Okay, so I say this leftover endosperm is used during seed germination. So when the seed germinates, so at that time, so it may use whatever that leftover endosperm. Okay, so that's what we see in case of albuminous seeds, which I will be discussing, okay, when I tell you about the seeds. Okay, right. So this is about the endosperm fate which is formed, right? It may be completely consumed or it may not be completely consumed. So let's see some examples under completely consumed. We see in case of right, peas, in case of ground nuts, okay, and even in case of beans, right, we don't find endosperm, okay, present in those mature seeds, okay. And now we see in the endosperm present still in case of castor and in case of coconut, right. So in them we shall see the remnant endosperm present, right. Hope this part is clear. So now let's see how, okay, embryo develops from the zygote. So here in this case I'm going to discuss especially, okay, embryogeny in dicotyledons. So in dicot plants, how the embryo develops from the zygote, so that I'm going to discuss. Of course, the point is that we know angiospermic, okay, plants, so which we classified into two types, okay, dicots and monocots, right. Of course, we know that out of the dicots and monocots, these are advanced angiospermic plants, okay, when compared with the dicot angiospermic plants, right. So that part we shall see it later. Now when we see the embryogeny in case of monocots and dicots, the early embryonic stages are similar, okay. So I say that early embryonic stages are similar in monocots and also in dicots, okay. So if I talk about the early stages like the pro embryo and the globular embryo, so these will be similar, okay, in both these cases, that is monocots and dicots. But after that the later stages may change, okay. Of course they will change in case of monocot plants, as they are advanced when compared with the dicot plants, right. So we know that the embryo develops at micropylor end. See, point is that when we studied the structure of embryo sac, we have seen the presence of egg, okay, towards the micropylor end, and of course the fertilization also happened there itself, and then that egg which is fertilized became zygote, and that is present, okay, towards the micropylor end only, okay. Now that zygote is going to divide, so, and it is present, okay, near this micropylor end. So here this is the zygote, okay. So say imagine already certain amount of endosperm is formed, even though I'm not drawing it here, okay. So there is a presence of endosperm, because as I already mentioned that, okay, certain amount of endosperm should form before the zygote start dividing and then resulting in the formation of embryo, because right, the zygote will not be, if it doesn't happen, then the zygote will not get the food, and it will starve, okay, right. So the zygote is present at the micropylor end, okay. Now so this zygote is going to divide by mitosis, so which I'll just mention here, okay. So in case of the dicots, the zygote divides and results in the formation of next stage called the proembryo, okay. So the proembryo later forms a globular embryo. The globular embryo later forms a heart-shaped embryo, so it later forms heart-shaped embryo, and then finally it will become mature embryo, okay. So these are the stages in case of dicot plants, Okay, so the zygote divides to form pro-embryo. The next stage is, okay, globular embryo. Then the next stage heart shaped, and lastly is the mature embryo. So you can see these are the various stages that we see, okay, during the development of embryo in case of dicots. Okay, so now let's see this zygote which is present towards the micropylor end is going to divide. Okay, and of course it has resulted in the formation of two cells. Of course all these divisions will be mitotic divisions. Okay, so from zygote to the mature embryo, all those will include only the mitotic divisions. So here it results in the formation of a basal cell, and there will be apical cell, right? So when there is formation something like this with the basal cell and the apical cell, of course now this is the next stage, what I call it the pro embryo stage, right? So this basal cell, of course later it forms, of course, the basal cell along with the suspensor, whereas this apical cell is going to develop into embryo. So the next stage is the globular embryo. So you can see in this case this is the basal cell, and on that you will find a suspensor. Okay, so here inside this I'm going to show you how it all taking place. Okay, so here you will find okay the presence of the suspensor cell, I mean the basal cell, and above that you will find the suspensor. So here a linear arranged cells, okay, will form the suspensor. So this is the basal cell, and this forms the suspensor. Okay, so this actually helps in pushing the embryo into the endosperm, right? And here you can see here this is okay the embryo, and you can see this embryo appears in a globular shape. Okay, so this is what I call it the globular embryo, is a globular embryo, right? So the first stage where okay we have seen the pro embryo, right, once the zygote divides okay into two cells, and then later the further divisions will result in the formation of a suspensor. Okay, so here showing inside the embryo sac. Okay, so the situation is like this. So this is the embryo is present inside like this. Right. So now once this is completed, the next stage, the further mitotic divisions will result in the formation of a heart-shaped embryo. Okay, of course the basal cell and the suspensor will remain same. Okay, right, of course the suspensor may become little elongated, I mean little cells get added. So this is the basal cell, and then okay the globular embryo will become okay so somewhat like this. So this is how you will be seeing. Okay, so a lot of divisions, and that results in the formation of okay many more cells. Okay, so you can see in this case the embryo appears in a heart shape, right? So hence this is what we call the next stage, the heart-shaped embryo. So this is the heart-shaped embryo, right? So the pro-embryo, the next stage which we have seen, the globular embryo. The globular embryo became the heart-shaped embryo, right? So this heart-shaped embryo, it's going to become into the, okay, the next stage, right? So what we call it the mature embryo, right? So here the basal cell, of course, this is the basal cell, and this is the suspensor, and this is the embryo that is formed. Okay, so here the last one, the last stage, so you can see the basal cell remaining, and the suspensor also same. Okay, and then, okay, so this one will be seen in somewhat like this, the development. Okay, right, so this is the basal cell, and this one is the suspensor. Okay, and this is the embryo, which with the two cotyledons, you can see, because I told that I am going to discuss about this embryogeny in dicots. So you can see there are two cotyledons, so these are cotyledons, okay, present, right? And this is what I say, the mature embryo. Right, so we have seen all the stages, the globular, I mean the first one, the pro embryo, then changing to globular embryo, and then the heart shape, and then, okay, the mature embryo. Right, so this is how the embryogeny occurs in case of the dicot plant. So this is the, the mature embryo. Of course, the same thing if I need to draw inside the, okay, so this, so you will be seeing okay, so this is how the embryo, that is the mature embryo, present inside that, okay, the embryo sac, of course, okay, which has become the endosperm, in fact, okay, embedded in it, right? So this is how the embryogeny in dicot. So with this I will end today's video. So in the next video I am going to discuss in detail about the dicot embryo, the structure of the dicot embryo, and we will also see about monocot embryo, and then later I shall start discussing about the seed in angiospermic plants. Thank you.